Good evening, everyone. My name is Peter Kariva, and I'm the CEO and president of the Aquarium of the Pacific. And first, I want to thank the sponsors for this evening lecture series, Gazette Newspaper and Courtyard Marriott. Well, tonight we're going to learn about corals and coral reefs and hear a little bit of a hopeful story, I think. You'll be seeing this in the lecture, but you probably already recognize that reefs are a big deal. You know, they are nurseries for fish, lots of variety of life, biodiversity. They dampen storm surge and protect shorelines. They're beautiful, a source of ecotourism. But a lot of my friends in the conservation business are depressed and demoralized because we're losing them at such a rapid rate. But tonight we're going to have Bart Shepard, who's from the California Academy of Science, and he's one of the co-directors of an initiative that's called Hope for Reefs. And he's one of a group of conservationists and scientists around the world who are not depressed and demoralized, but are taking action to see what they can do to restore reefs. You know, it's conventional to talk about the speakers' degrees and honors and all that, but you can look up his biography. What I found most interesting is at the age of 6, he started with an aquarium with tropical fish in it, and there he was, age of 6, set on the path of his career. At the age of 6, I was going to be an outfielder for the New York Yankees. It didn't work so well for me. So take it away, Bart. I'm really looking forward to hearing what you have to say.
Great, thank you, Peter. And you know, the qualifier there is I did have a short time where I thought I was going to be a rock and roll guitar player, but that didn't work out for me either. But anyway, I'm glad to be here virtually with you all today to talk to you about a program that I lead about saving animals from extinction through the Association of Zoos and Aquariums. That's working really on restoring coral reefs and bringing some, as Peter said, hope for the future of coral reefs. But first, let's talk a little bit. You know, this is the Verde Island in the Philippines. I was lucky enough to go there a few years back, and it is absolutely one of the most gorgeous and beautiful things I've ever seen in my life. We have this remarkable, healthy coral reef. It's just loaded with all kinds of colorful fish. And all of this is made possible by these incredible little tiny animals, coral polyp, that harnesses the energy from the sun using an algae that they grow inside their tissues, and they clone themselves asexually. They just bud into 2, into 4, and then they take dissolved minerals from the ocean and they make limestone structures. They basically build rock from water using the power of the sun. And it's a remarkable thing that creates a tremendous amount of habitat and is responsible for a lot of the biodiversity that we have in the ocean, you know, as Peter alluded to. Coral reefs occupy less than 1% of the world's ocean floor. And it's actually far less than 1%. And yet they harbor more than 25% of all marine life that we find in the oceans. And some scientists estimate that could even be as high as 30%. As Peter mentioned, they provide a human society with a great deal of natural capital or ecosystem services, including, you know, a tremendous amount of annual tourism revenue. And, you know, in a normal world, many of us would pack up and go and visit a tropical island like this, like Guam, for a week or so of vacation in the sun, snorkel and enjoy the proximity to coral reefs. And people who live in these types of locations year round often derive a great deal of their food directly from the reefs. And a healthy reef ecosystem can produce a tremendous amount of fish as food for the people that live near it. And as we're seeing in the news this week with hurricanes just hitting the Gulf Coast, you know, this is a tremendously damaging force that comes into our cities and into our civilization. And coral reefs have the ability to reduce the wave energy that's associated with storms and storm surge. And so they provide all of these benefits to us and are important on their own right for being diverse and beautiful ecosystems, but also for these ecosystem services, this natural capital that they provide. Now, I know probably many of you watching, if I were to ask you to picture a coral reef in your mind, you might picture something like this, right? And this is a typical shallow water coral reef. This happens to be in Ponape in Micronesia, and it's again a beautiful, diverse environment. And most coral reefs that we've been studying that we know about are limited, we thought, to these shallow waters, to maybe 100 or 150 foot deep. That's largely due to the limitations in technology that we've had available to us, scuba diving. And scientists who go scuba diving and study coral reefs are limited by the equipment that they have, and often the universities and the programs that they work for to going any deeper. And I've been fortunate enough in recent years to study deeper reefs, what we call mesophotic coral reefs, which are middle light, or the twilight zone, it's often called, which extend down to about 500 feet deep. And what we're finding is that these are unique and diverse and beautiful habitats. This happens to be in the Philippines, and this is about maybe 150 feet deep. And there are these wonderful soft corals that come up out of the bottom, get into the current and filter feed from that environment. Again, it's very different from those shallow reefs, unique species not found in the shallow environment. You go deeper. This is in the Cayman Islands. This is at about 100 meters deep, so 350 feet deep. And it's dominated by these large, colorful sponges and these whip corals, these black corals. So unique assemblages of animals that you don't see on the shallow reefs. So we're learning that coral reefs actually go much deeper than we expected. They're unique down there. They are threatened down there, and they are equally in need of protection and conservation as those shallow water reefs. And I love this photo because it really summarizes kind of why reefs are important. This is in the Bahamas, and you have this tiny little patch reef in a big sandy area, a wide open sandy area in the Bahamas. And where are all the fish, right? They're all clustered right around that structure that's been built by these little coral animals. And it shows, that these animals and all of this extra diversity that exists on coral reefs is reliant upon that healthy reef structure. Now, unfortunately, this is what you tend to see more often than not. This is in the Cayman Islands, and this is, a largely dead and denuded reef, and there's very few fish or anything there, right? And we're seeing this occur more and more all around the world with a number of different ultimate reasons. And the main problem with this is that there are these nested types of impacts that are happening on coral reefs. You have global impacts that are impacting all coral reefs all around the world. You have regional impacts that are impacting certain places, like the Caribbean, for instance. And then you have local impacts, which may impact one reef or one island or one tiny little area. And if we kind of dissect into these and look at what some of these causes are, you see the main one is carbon emissions and ocean warming that is part of and parcel with climate change. Also having major impacts are coastal development, different fishing practices that aren't sustainable or responsible, and pollution, runoff, sewage, plastics, and things like that. Now, sometimes these declines can be very dramatic and occur in a very, very rapid time frame, and it's hard to not be, depressed and disillusioned by these things when you experience them firsthand. These are two photos that were taken in French Polynesia about 10 weeks apart, and I was on the left-hand side there in Tahiti with a group of scientists that were studying these deep reefs, these mesophotic reefs. And when we do that, we're using technical diving, and we have to come up to the surface, and we stay in the shallow for hours at a time, right? So 3 hours, 4 hours, something like that, decompressing, depending on the dive that we made. And in Tahiti, we were tremendously and very much surprised, pleasantly surprised by the diversity and by the health of this ecosystem, given that it was Tahiti, which is, a major port and has a huge population and a lot of cruise ships coming in and out. And, we found these relatively diverse, healthy, shallow environments that we were spending hours swimming over as we were decompressing from our deep dives. 10 weeks later, we returned and we went to Moorea, which is just one island over. You can see them from each other, very close. And we found what was on the photo on the right. And in the interim, in that time frame, there had been a warming event where the ocean had gotten very hot around the French Polynesia, and the corals massively bleached. And what that means is they expel that algae that lives within their tissues. It gives them the energy that they need to live and grow. They basically spit it out because it's a breakdown in that sort of relationship that's happening. And we saw this massive bleaching over a huge area, a huge geographic area on Moorea. It was incredibly depressing to have to swim over this for like 3 hours or 4 hours a day watching this. And then by the end of the 2 weeks or so that we were there, a lot of these corals had already died and were starting to have algae growing all over them. So this is a direct result of carbon emissions, climate change, and ocean warming. And the breakdown of that relationship between the coral and the algae that sustains it occurs when the water gets hot and stays hot. Now, an example of maybe a regional level of threat to coral reefs is this from the Caribbean. This is in the Cayman Islands, and you can see, the reef itself is rather poor in terms of the coverage of corals. There's a fair amount of algae growing all over it. There are some corals that show evidence of prior struggles with disease or maybe bleaching. And then you have this invasive species, the Indo-Pacific lionfish, swimming there in the front. And that is a fish that was introduced into Florida from an aquarium and has now spread throughout the entire Indo-Pacific basin. And so, again, spread across the entire Caribbean basin. And so an impact that's on a regional scale. And we also see other regional type impacts like this. This is a crown of thorns starfish. And these will bloom periodically, and they're a coral predator. So they crawl across the reef and they just digest every coral that they come in contact with. And when we were in Ponape, Micronesia a couple of years ago, we were caught in the middle of bloom of the crown of thorns. There were dozens of these things all over the reef eating every coral that they came across. And something like that is probably cyclically occurs in a natural cycle. But when you compound all of these threats, these local, regional, and global threats, and you have the pace of those continuing to increase, it doesn't give the corals time to recover. As an example of a local impact of coastal development, here you see an airport that was built on top of a coral reef. And this is in Micronesia. This is either Kwajalein or Coast Rai. It's one of the stops on the United Island Hopper flight, which pops around the Pacific. And again, it's understandable. It's a forested mountainous island. Where can you build an airport? They built it on top of the coral reef, and that's probably an impact that will last for decades to come. And in the Philippines, we see similar local impacts. This is a small village that you can see built out on this tiny little spit of land. And there's a whole lot going on here, right? You have this little village almost certainly has no primary wastewater treatment. So all the sewage, all the waste from that village is going right out into the water. They're clearly struggling with either sea level rise or more powerful storms because they've sandbagged the entire back end of that village. The beach is covered with plastic trash and litter that's washed down from the river and off of the land and collected on the beach. And yet there are 3 or 4 people out there wading in the water, waist to chest deep with nets, collecting fish to feed their families in that village. And here's another example in the Philippines. This is a lagoon off of the coast of the Bolinao Marine Laboratory of the Marine Science Institute of the Philippines. And there are all these structures out there in the water. And if you look at them up close, you see these are net pens to raise milkfish, which is an important food fish in the Philippines. And so there'll be somebody that goes out and lives in this little hut, and those pink bags there are all filled with pellets, fish food. And that person will feed the fish day in and day out until they can grow them to a market size, and then they'll be pulled out and taken to market. And we were able actually to dive right nearby there, and it looked like this. And this is not really what you would picture when you think of a tropical coral reef, right? This, to me, looks more like the Northern California coast where I am right now in the summertime. You have this phytoplankton bloom, the water's kind of pea green, and there's all this fluff that's floating around in the water column. That's bacteria and detritus and waste from those fish pens that's nutrients that are suspended in the water column. It's remarkable that corals can exist in this environment. And another example of a local scale impact, this is a typical anchor used by dive boats in places like the Philippines and Indonesia. And if there aren't mooring buoys for where the recreational divers are diving, the first mate of the boat will just throw this over onto the reef, let the divers out, divers will do their dive. At the end of the dive, they pull the anchor up. The next day repeats. And so you can see how multiple dive boats hitting the same spot on the same day, all dropping anchors. This is a small scale impact that is compounded over a longer time, and it doesn't give the corals time to recover. And as we go deeper and start to look at these twilight zone reefs, these mesophotic reefs, we see the same kinds of impacts. Impacts of fishing, anchor lines, impacts of plastic and garbage. And this one is, I love, there's a fishing line going across with a little sea urchin that's crawled up on the fishing line, and the sea urchin's trying to camouflage itself with a piece of plastic trash. Right? So public aquariums traditionally have tried to address the coral reef crisis in these ways. We have exhibits, we do education programs, we do what we call public engagement. We interact with people and we tell them the stories of coral reefs and the challenges that coral reefs are facing, much like I'm trying to do right now. And at my facility, we have a Coral Reefs of the World exhibit, which tells all these stories and why coral reefs are important. We put a diver into our large Philippine coral reef exhibit. That diver goes down, has a conversation with a presenter on the dry side. School children and visitors are there. They can ask questions of the diver, get information about coral reefs. And even, in my facility, we have a planetarium show, and we put together a show called Expedition Reef, which does this. It creates a digital virtual reef, takes people down onto that reef, and tells them these same kinds of stories. But more and more, we're seeing new roles for public aquariums, roles that are emerging around the research and conservation and even restoration. And that's where I want to spend the bulk of the rest of my time talking about is the work that we're doing in terms of restoration and renewing coral reefs and making them better than what we have found when we went out there ourselves. And in order to talk about that, I'm going to use one of my favorite animals, the majestic and charismatic Caribbean elkhorn coral. And to me, there are very few animals on the planet that are as beautiful and as remarkable as this. Here you have this coral with its arms outstretched, taking in the rays of the sun, converting that energy, using the algae in its tissues into rock, building these massive structures that surround Caribbean islands. This is in Curacao, where there are still some healthy stands of elkhorn coral, and it's a wonderful place to go and visit. Historically, elkhorn coral would ring Caribbean islands with these massive structures, what we call monospecific stands. It's a single species of coral filling up basically all of the space as far as you can see. And I can remember being a young kid, maybe 12 or 13, and being in St. John in the US Virgin Islands and going out and snorkeling and coming across something like this and trying to figure out how I could get over to the other side of it because I knew all the cool stuff was on the other side of it. But these branches extend right to the surface of the water. Often when waves go by, tips of the branches are sticking up out of the ocean, and they ring Caribbean islands up until the 1970s or 80s. And even if you look back at what some of the colonists wrote when they first came over from Europe, there's accounts of how it was difficult to get to the land on some of these Caribbean islands because of this ring of reef that surrounded each island. And that was elkhorn coral. In the 1970s and 80s and going into the 90s, there was a disease that went through and wiped out a lot of the elkhorn coral, unfortunately leaving behind this. And this is Buck Island in the United States Virgin Islands. This is a United States National Park. And it is an elkhorn coral graveyard. You can see the skeletons all piled up on each other there. Again, the branches reaching off right to the surface of the water, but all being broken down now into rubble and into sand. And even when you do find healthy elkhorn corals, you often find them off by themselves, like this individual in the Cayman Islands. One small colony out in the middle of a desert all by itself. And that essentially isolates this colony from any sort of reproduction, right? Corals, they spawn together in groups. and they need to have their neighbors all spawning at the same time in order for it to work. And so this coral being out by itself, away from everything else, even if it does produce gametes and spawn, there's nobody else there to fertilize it. So it's reproductively isolated. And in fact, we see no new baby elkhorn corals anywhere in the Caribbean. The reproduction has been shut down because the population declined to such an extent. So in response to this, a few years back, I led an effort to start a program for the Atlantic Acropora corals under the umbrella of SAFE, Saving Animals from Extinction, which is a program, an initiative by the Association of Zoos and Aquariums. And there were a number of accredited zoos and aquariums that joined us in this effort to save this elkhorn coral. And I have members of a steering committee from each of these that helps put together this program that I'll be talking about here. That we actually chose to focus on two species, the elkhorn coral and a close relative, the staghorn coral, both of them occurring in the Caribbean and both of them suffering from that same disease that took place a few decades back, leading to declines in their population of up to like 90 to 95% and causing them to both be listed as critically endangered on the IUCN Red List and listed on the United States Endangered Species Act. And that the latter piece there, being listed on the ESA, is important because then that mandates a federal recovery plan. And NOAA has put together a recovery plan for the Atlantic Acropora corals, elkhorn and staghorn corals. Our program, the SAFE program's goal is to support the recovery program, the federally mandated recovery program. And we do that through a variety of different ways that I'll talk about here. We want to propagate and outplant corals. We want to make more of them and put them out on the reef and boost those populations that are found out there in the wild. We also want to create living gene banks, or ARCs, and some of those will be on land, keeping the corals safer from some of the impacts that we've been talking about that are occurring on wild reefs. Nutrient input, habitat destruction, temperatures from climate change, disease, all of those things. We can control those in a public aquaria in our aquarium systems. And then we also wanted to support research on restoration. How can we improve the techniques that we have to regrow reefs, to renew reefs, to bring reefs sort of back to a point where they existed at some point in the past, where they were healthier and more vibrant. The problem with restoration right now is that restoration efforts for corals are tremendously limited in scale. And so let me kind of orient you here to this a little bit. On the bottom is basically just surface area, going from small to large. And then each of those boxes represents some of the types of impacts that we've been talking about earlier in this presentation. A ship grounding is a very small impact. Freighter or a commercial ship of some sort blows off its anchor or loses an engine and drifts into a reef, it causes a small scale damage. You move up into that wide range of human impacts around a square kilometer, maybe that's that airport. Maybe it's a sewage output somewhere on an island. Then you get into these regional impacts like the crown of thorns outbreaks, like these bleaching events that are affecting a geographic region. The challenge is that restoration is limited to about 1 hectare with our current techniques. So if you want to do something that is at the scale of the decline that coral reef is facing, we don't have that technology yet. We're not there yet. And so that's something that my program and my organization under the Hope for Reefs initiative here at the California Academy of Sciences set forth to change. And one of the ways that we can address coral reef decline, and particularly in the Caribbean, is through asexual propagation of corals and then outplanting. And what I mean by that is basically we can use kind of greenhouse gardening techniques. We can take a branching coral, we can break it up into a bunch of small pieces. We can grow each of those pieces to be bigger, and then we can plant those out back onto the reef and let them grow. And this work is being conducted to a very good scale by our colleagues in Florida, at the Florida Aquarium, at Mote Marine Laboratory, and Disney's doing work as well in the Bahamas. And there's a couple of challenges here. And I don't want to discount this technique because it is certainly a tool in the toolbox. And at this point, with the state of coral reefs and on a global scale, we need every tool in the toolbox, right? Sometimes you need the screwdriver, sometimes you need the hammer. And so this is one of the tools that we have available. But there are some challenges here. The first, of course, is genetic diversity, right? So you're creating clones, you're creating individual colonies of coral that are all genetically identical. If you take like the Coral Restoration Foundation in Florida does, a colony, you break it up into small pieces, you hang it on these mid-water nurseries, you grow them for some time, you break those up into small pieces. You can make thousands of these corals, but they all descend from that one founder individual, and they're all genetically identical. If you plant all those back out into an area, and then maybe there's a disease or some impact that we don't know about that's going to come down the line that these corals are susceptible to, you'll lose all of them because there's no natural genetic variation in that population. They're all clones, they're all genetically identical. And so that's a weakness to that approach, in my opinion. The other challenge with this is, of course, the limitations of labor. So here you have a scuba diver. She has expensive equipment. She had to be trained. She can take a coral fragment. She goes down on a dive. She has to find a suitable location. She scrubs the rock off with a brush to get the algae off. She uses some sort of epoxy putty or glue or a fastener to adhere that coral. And she can only do a certain number of those on a given dive. And while we're able to scale this using some sort of community-driven response and community outreach, working with local dive clubs, working with even veterans groups, having public aquarium professionals fly in and help with this kind of effort, we're still limited by the expensive equipment, the time that it takes underwater to do this on a meaningful scale. So a few years back, we started talking about, how could you take a different approach? How could you, like a farmer scattering seeds in a field, simply toss corals out onto a reef where they would attach themselves and grow and into the next generation? And in order to accomplish that, we started working with a group called Seacor International. Now, Seacor stands for Sexual Coral Reproduction. And we believe that the key to scaling restoration is by working with the coral's natural reproduction, by harnessing the spawning that occurs on coral reefs to collect gametes, cross-fertilize those gametes, and create entirely genetically new individuals that can then be outplanted back onto that reef. So in order to understand how that works, we have to talk about coral sex. You have to see coral sex in action. And this is what it looks like. It's kind of like snow in reverse. Right? You have, this is a colony of a cropper coral. This one's actually in the Philippines, but it has brooded these gamete bundles inside its body. It's grown sperm and egg bundles together. It's put them out at the surface of its tissue, at its mouth, and it has waited for this certain moment in time that is cued by an annual temperature cycle, and then the phase of the moon, and then the sun setting. And at that point, all of the corals in that environment will synchronize their spawning and they'll release the gametes into the water. And it looks something like this. They're all floating about, they're mixing and floating around in the water. And we actually stumbled upon this in the Philippines on a night dive. This was back in 2011. And it was a highlight of my professional career. We had no expectation that this was going to happen. We just sort of came up, and then all of a sudden there was all this stuff in the water, and we were like, what is it, all of this? And then when we realized what it was, we were literally screaming at each other through our regulators as we were witnessing this massive coral spawning in the Philippines. So let's break that down a little bit into this individual life cycle. You've got a spawning adult. This is a broadcast spawning, and there's other coral reproductive strategies, but for the purposes of this, we're just going to talk about broadcast spawning. They release the sperm and egg bundles from the polyps. They go up into the water, and they're buoyant, as you saw in the video. They float up to the surface, and they mix on that surface water and undergo fertilization and the development of the embryo. That embryo grows for a while and then becomes a passive planktonic larvae. It's just drifting as part of the plankton on the surface of the water for a few days, maybe. And then at some point, about a day later, it becomes this active planktonic larvae. And this to me is one of the most remarkable things about coral reproductive biology. I mean, here you have an animal that, most people probably think is a rock. It lives attached to the bottom of the ocean and can live for thousands of years, cloning itself and growing over and over and over again. one point, maybe one day in its life, it has this phase where it's actively swimming and exploring the environment, searching for a place to settle and become a new colony of coral. And that to me is just remarkable. Undergoes settlement, it starts to metamorphose, it starts to grow the skeletal structures of a coral, it buds itself, cloning itself over and over again, becomes a recruit, and in time would grow up and survive and spawn and complete the cycle. So in order to harness the coral's life cycle, in order for us to do restoration, we had to look at managing each of these different phases. We had to find a way to collect gametes on the reef. We had to find a way to culture that larvae through these different phases, when it's just passively drifting and to when it becomes an active larvae exploring its environment. We had to come up with something to settle that larvae on that would allow us to get it back out on to the reef, to outplant it back onto the reef. And then we monitor those techniques and improve our methods over time. Now, fortunately, public aquarium professionals are incredibly resourceful, right? They often work with tight spaces, with limited budgets. They can build anything out of like PVC pipe and some salad bowls and maybe some plastic light diffusing material. Very creative, very resourceful people. So they're a wonderful group to partner with academic scientists and conservation practitioners. So they bring a lot to the table. So let's walk through sort of the methods that we've been using. Here's our old friend, the Caribbean elkhorn coral. And maybe you can see on some of the branches, it's a lighter color, maybe a little more pink. Those are the branches where the gametes have set. It's pushed the sperm and egg bundles up to the tip of the mouths, and the polyps are holding on to them, waiting for that moment in time when it's synchronized, and they'll spit those gametes out into the water column. So we come in at night and we wrap those branches with collecting nets. And this is like the hairiest diving you will ever do. You're at night, it's a high surge environment. You're literally, a foot or so from the surface of the ocean, and waves are crashing over top of you, and you're trying to wrap what's essentially a pillowcase around the branch of a critically endangered species. It's super nerve-wracking. But you do that, you wrap these nets around there, the gametes start to float up through the nets and into these collecting tubes, which we can then bring back to the laboratory and cross-fertilize. And as I said, public aquarium professionals are very resourceful. Here's the preferred technique for conducting fertilization. You have a turkey baster and a fat separator like you would use to make gravy on Thanksgiving. And you mix all those things together, sperm and the eggs from different colonies. You ensure good fertilization, and then you culture those larvae. In this case here, they're being cultured in these clear plastic tanks that are constructed out of plastic salad bowls. And they have a special current that sort of blows around in there that keeps the corals up and towards the middle of the water. They're floating on the surface right in the middle, not contacting the walls, much like they would be out in the ocean. And you culture them this way for a few days until they become those active swimming larvae. And at that point, you have to shift your approach, and you have to look for ways to settle them. And here you can see some active swimming larvae in a few drops of water on a dollar bill to give you a sense of the scale that we're talking about in terms of those little free swimming, what are called planula larvae. So here you'll see this is a small rock that we have given them as a place to settle. And you'll see the one that's on the lower left there is exploring its environment, and it's found a spot that it's very intrigued by. It's in a little groove. There's a little like cut in the rock there, and it's right on the margin of this sort of pink algae, this what we call coralline algae. And those things seem to be cues. And with scientific study, we've been able to predict where corals will settle to help with these restoration efforts. Here's another shot of on the bottom, you have a planula larvae that's still sort of elongated and explores its environment. And then above it, you have two that have begun to settle and undergo metamorphosis. And maybe you can see there are these faint lines in there that are kind of like asterisks. And those are what we call the septi, the radial skeletal structures that come out from that central mouth and go out to the margin of the colony. Again, where are they? They're right next to that pink and purple coralline algae. They're settling there, and they're choosing that spot. They'll grow a little bit. They get these nice little stubby tentacles that are characteristic of stony corals. They begin to clone themselves over and over again, and they become a recruit, a coral recruit. And here you can see this one is in an artificial substrate. It has a groove of a specific width that was manufactured. And again, that coral chose a spot in that groove right next to that pink coralline algae, and that's preferential. And by understanding what the keys to settlement are and being able to control that and make them settle in a specific spot, that opens up all kinds of opportunities. That gives us the ability to create substrates, seeding units, that we can use to simply scatter onto the reef from a boat. They'll fall down. They're kind of shaped like jacks or tire spikes or caltrops. So one point is always pointing up. Those grooves ensure that the corals are in that one of those points that are pointing up. And it gives us the ability to upscale restoration. Here's a seeding unit that was settled in Guam, and I'm happy to say the Aquarium of the Pacific participated in this work in Guam a few years back, working with a scientist, James Guest. And you can maybe make out the shape, that triangular shape that's in the center. The seeding unit stabilized itself into the environment. It delivered a coral onto that reef environment. So we started working with different designs of these seeding units, trying to optimize not only the way that the corals settle on them in a predictable way, but also the seeding unit's retention in the environment, right? When we go back 3 months later, can we still find them? Have they been swept away by the waves, by the wind and storms, or buried by sand, or are they found still? They stabilize themselves in an environment. And we started with these designs, and then through a partnership with SECOR and with the California Academy of Sciences and Autodesk Foundation, and whose Autodesk Foundation is dedicated to using design to solve some environmental problems. We worked on this next generation of seeding units, and these are different designs that are in test right now. These are actually 3D printed in ceramic, and they're being field trialed now to see which ones work best, again, in terms of where corals settle and can we predictably settle corals on them in the right places, and what's the retention in that environment, that natural environment. We also had to upscale our plastic salad bowls because if we want to deal with corals in the tens of thousands or hundreds of thousands at a time, we can't do it in simple systems like that. So we created these we call cribs, which are coral rearing in situ basins. It's essentially a floating pool with mesh sides that lets water in and out. And so instead of growing these coral larvae in land-based systems that are an aquarium that you have to manage, here you're using the natural ocean water, and you then introduce the seeding units in there for the corals to settle. Now we know this technique works, and that's really the great piece at the beginning is we know that we can bring corals back out onto a reef and have them reproduce with their wild counterparts. This paper that came out a few years back documents the first lab-reared elkhorn coral spawning with its wild counterparts. And I'm not an author on the paper, but I was one of the people that helped collect the sperm and eggs that were used to grow this coral and many others that were planted out at that same time. And 4 years after they were planted, that coral spawned with its native population in Curacao. And our ultimate goal here is to get to something like this. That we could come into an environment with a new partner or a new collaborator, and we would do an assessment of the reef, like you see on the left. And what's the status of that reef? Is it healthy? Is it degraded? Is it somewhere in between? And then we would look across the bottom. What are the species that can exist in that type of environment? Certain corals, like the elkhorn and staghorn coral, can only survive in healthy reef environments. Others are more resilient and can survive in all kinds of different environments. And so you pick the species for the environment and the status of that reef, and then you apply the best methods for that species. So we're looking at creating something like this. We are also looking at diversifying our program and beginning to work with many other species. There are other endangered species in the Caribbean, and now they're with an emerging disease. There are threats to a great many other species of massive corals and brain corals that aren't really necessarily suited towards the techniques that I've been talking about here. So we've got some other techniques that the program has been doing that are being led by project partners. And the first of those that I'll quickly talk about is microfragmentation. Now, I mentioned in the beginning asexual propagation and fragmentation. That works really well for these branching colonies. You can go out there, you break a branch, you grow it into a new colony. You can break that one up into 2 or 3, you grow that into a new colony. This doesn't work so well for the branching colonies. It works great. Microfragmentation is an approach that allows us to use this on massive corals, right? On big boulder-like round shaped corals that aren't branching. And so in the microfragmentation approaches, you use like a tile saw, and you cut these things into tiny, tiny little pieces, and then you mount those pieces next to each other. And for some reason, that elicits this rapid growth response where those corals, they start budding and cloning themselves, and they try and scan over that space in between them. And so you're able to very quickly grow new colonies from these tiny, tiny little pieces that you take. And it works well, as I said, with these massive corals, with these boulder corals. And it's being done with Mote Marine Laboratory with a great many species. And they just had a great breakthrough just about a month ago where for the first time they documented a coral on a reef in the Florida Keys that they had outplanted using this microfragmentation technique, and it had spawned with its wild counterparts as well. So we know, again, that the technique can get corals out there that are become reproductively active. I also mentioned in the beginning a focus on creating land-based nurseries, genetic arcs for holding these corals outside of the wild environments where they're facing many of these threats. And Florida Aquarium is a leader in this. They have these greenhouses at a Center for Conservation, and they can track genetic strains, maintain them in a stable environment, propagate them, use them for restoration, and maintain an arc of corals on land that are protected and separate from a lot of the threats that are occurring to wild reefs. It also opens up new opportunities for research and restoration techniques, and Florida Aquarium's had some great success with that as well. This is a land-based spawning system for another endangered species of coral called pillar coral. And what basically is done here is using artificial lights and temperature control, you mimic the environmental cues that elicit that spawning that we talked about in corals. Seasonal variation in water temperature, the phases of the moon, the sunrise and the sunset. And Florida Aquarium has been successful now 2 years in a row in eliciting spawning in pillar coral, providing gametes and larvae to be used for restoration purposes, for research, and is a great advance. And several other organizations now have invested in spawning systems. We have a small facility here at the California Academy of Sciences where we are spawning some Indo-Pacific corals, and Mote Marine Laboratory is in the process of building a spawning facility as well. All of this, I should say, came out of a small museum outside of London called the Horniman Museum, and there's a scientist there by the name of Jamie Craggs who pioneered this approach. And then another aspect of our project that is causing us to diversify the species that we're working with within this SAFE program is an emerging disease throughout the Caribbean basin that's known as SCTLD, stony coral tissue loss disease. And this disease started around Miami, and it has spread both north and south on the Florida Reef Tract. It is hugely destructive. It impacts more than 25 species of corals with almost 100% fatality. We don't exactly know what it is. We think it's a bacteria, but it has now been documented in Mexico, in the Virgin Islands, and in the Bahamas. And so we know it now is likely to become a regional disease threat that will impact the entire tropical Atlantic. It looks something like this. You can see here, this is a brain coral. On the front side of it is sort of this healthy golden brown color. Then there's a section of bare white skeleton, and then some skeleton that is starting to be overgrown with algae. And in a very short period of time, this coral will be completely consumed by this disease and will be killed. And so zoos and aquariums across the United States have stepped up in partnership with the Florida Fish and Wildlife Conservation Commission and NOAA, and we've launched a Florida Reef Tract Rescue Project to get some of these corals out into these land-based nurseries, to hold them into biosecure aquarium facilities where we can let the disease run its course, maybe figure out treatment options, and then perhaps plant these corals back out onto the reef in due time. And currently there are more than nearly 2,000 corals at about 19 different facilities. So I hope I've given you a snapshot of the ways that public aquariums and zoos are stepping up to help address the coral reef crisis through restoration efforts and through these genetic arcs and by supporting research. And the Association of Zoos and Aquariums put out this infographic a couple of years back, showing that there have been 30 facilities that have invested $3 million in coral conservation between 2014 and 2016. And about half of that was with those two Atlantic species, the elkhorn and staghorn coral that I've been talking about. And I've been tracking this data since then. And if you look at the magnitude of that conservation and research effort, you can see it has continually grown and is now over $3 million in the last year in 2019, devoted towards addressing coral reef decline. And 2020, and I've heard it said it's unprecedented is an overused word, but 2020 is certainly an unprecedented year. We will see how this impacts our programs, but my hope is that we continue to value conservation of coral reefs, and we continue to put forth our best efforts to make sure that reefs have a future on this planet with us. With that, I'd like to say thank you, my colleagues here at the Academy, my SAFE Coral Program partners and collaborators, and of course the donors and supporters of the Hope for Reefs initiative, including Autodesk Foundation and Kingfisher Foundation, who've made all this work possible. And if you're interested in learning more, I'm fairly active on Twitter. You can follow me at SteinhartBart and follow the website at calacademy.org. Thank you.
Great, thanks a lot. That was terrific, Bart. Do you teach at a university? You'd be a great teacher because it was so easy to follow, and it was outstanding.
Yeah, thank you.
It gets me thinking. You at the end, you showed that graph of you're getting up to $3 million a year, but I gotta believe to really take this to scale, to really make an impact, given the global nature, you're talking about something that would cost $3 billion, not $3 million. I mean, is that right?
No, absolutely. Those contributions are just of the Association of Zoo and Aquarium accredited members. So that's a small part of the work that's being done and reef restoration on a global scale. But you're absolutely right. We need broader partnerships. Each of these locations needs to be community-driven conservation. You need to have invested stakeholders in the local community. And we really need the investment and the support of major industry, things like, the insurance industry, who has to pay out every time a hurricane passes over a reef and wipes out an area, right? Those are the kinds of investments that we need in that scale.
Like, you showed those really neat those tri-tip things that you put out. That how much does just one of those cost?
We've gotten those down. Initially they were all handmade with cement using these urethane molds and, one-on-one handmade. And at that point, they're like dollars, in terms of time and labor. But we've gotten them down to less than $1. I want to say they're about 50 cents each now. And I think we can get them down cheaper, with mass production.
I've seen articles in conservation in general about the total cost. Has anybody taken a crack at, like, what global investment would it take to save coral reefs with the existing technology? Has anybody, done that back of the envelope calculation?
Yeah, I don't know that number off the top of my head, but I know there are folks that are scientists and, probably former colleagues of yours from the Nature Conservancy who have worked on that and that very question and could give a better estimate.
I mean, it would be a huge number, I'm sure, but, as you mentioned in the beginning, if you wanted to estimate the economic value of corals, it would be on the same scale as well.
Yeah. Oh, absolutely.
So
absolutely.
it's not some of the other approaches being used, and I wonder if you could comment on them. Is one is assisted evolution, where the idea is you assist evolution and then the species do it on their own, and the other one is genetic engineering. In a lot of people don't recognize this, but in conservation in general, gene editing and genetic engineering is increasingly discussed as a conservation strategy. The American chestnut is a good example, which is virtually wiped out, but genetic engineering could help restore it. What tell us a little bit about assisted evolution and genetic engineering and your thoughts.
Yeah, no, I have a lot of thoughts about that. I mean, I think regardless of your opinion of the the approach, you need a delivery technique, right? So using the culture methods and the seeding unit approach allows us, even if it's used for assisted evolution or not, it's a technique that is valid, a delivery technique. I think back, I had a conversation with Dr. Ileana Bombs, who's a coral geneticist from Penn State, a number of years ago about this very subject. And there's been a lot in the press about supercorals, right? And like, can we engineer or breed supercorals that overcome that are able to overcome or are resilient to these threats and the stressors that are out there right now in the wild. And her take on that, which I thought was very insightful, was, evolution has already done that for us, right? When you force a species through a bottleneck where 90 to 95% of the individuals die because of disease and warming and habitat destruction, the 5% that are still there, those are supercorals. We already have them. The problem is that they're not reproducing effectively because they're too spread apart. They don't have the density anymore that allows for successful reproduction. So our approach really has been looking at interseeding in that way and helping those corals with reproduction. So maybe it's more assisted reproduction, instead of assisted evolution. I also think that assisted migration is another concept people have talked about. Can you move corals to environments now where, because the ocean's warming, maybe it was a space where they couldn't exist prior, but now might exist. And I think that's worth considering as well. Humans have engineered and modified and managed ecosystems all over this planet. What's stopping us from doing that on coral reefs?
That's a really good point you make about evolution, because as an evolutionary biologist, the rate of evolution is proportional to the strength of natural selection, and that's what you've just said. When you have that type of mortality going on, you're accelerating evolution. I'm wondering if there's, with this pandemic, ecotourism is shut down, sort of travel to small islands and things like that are shut down. There may be a reduction in sewage and pollution like that. Are there any stories emerging like there are on the land of seeing recovery or changes in the ecosystem just because of the suspension of human intrusion? Or have you heard anything about that in the coral?
No, not in the coral world. You see things in the news about animals that are out in environments behaving differently than they were. I think with coral reef decline, and these nested threats of global and regional and local threats, maybe you would see some relaxation of the local threats, but the larger threats are still there. There's still the same amount of carbon dioxide in the atmosphere. There's still the same amount of plastic in the ocean.
Yeah, I know this is, but coral reefs are the basis of an economy for a lot of small islands. And I've worked with the Waite Foundation, who really has the brilliant idea of working with small island developing states because they really have political leverage. But those islands must really be getting just nailed by the pandemic in terms of their economy, and at the same time the coral reefs that draw in the tourists are hurting. Is there a sort of a global recognition, not just among scientists and the conservation, but like World Bank and institutions that care about economies? Is there any awareness of the issue?
I don't know. I don't know the answer to that. I think it's an excellent point in that, it may directly impact a bunch of people on an island, but the health of these reefs and the health of the ocean impacts every one of us. And so It does require a global level awareness and a global level response to really address some of these challenges.
So I just want to, end with one question that, I often get asked when I talk about conservation, and it's a hard one to answer. Is, you show those beautiful slides, and you make people care. Are there things that an individual could do? Are there things that the people that have just watched your terrific lecture? Are there things? Is there anything they can do to be part of the solution?
Yeah, absolutely. I mean, I think this year more than anything, what they can do is vote, right? I mean, we need leadership
Yes.
at our national level, and we need leadership on a global level that recognizes that climate change is real and that it is the greatest existential threat that is facing our society, perhaps in thousands of years, and that we need to all work together to mitigate that. You can eat sustainable seafood, you can buy a plug-in car, you can change your light bulbs to LEDs. All of those things are great, but the crisis will not be addressed until we have a government that steps forward and wants to address it.
Well, I think that's a great point to end on. It's very trendy to have lifestyle choices that are pro-environment, but in the end, the scale of the challenge you've laid out really requires global and national action. Thanks a lot for spending the evening with us.
My pleasure.